Hi everyone, myself Sundari Ministry from CSA third year. So as a part of the machine learning and techniques laboratory, let's look into experiment number 8, which is nothing but self-organizing maps. So first we look into what is self-organizing maps. Self-organizing maps, often referred to as kernel map or self-organizing feature maps, is a type of artificial neural network used for unsupervised learning. Unlike supervised learning, SOM trains without unlabeled data. This is nothing but it trains without a labeled data, without valid input and output features, discovering inherent groupings or patterns on its own. So let us look into the experiment. So first we are going to import the necessary libraries, pandas, numpy, matplot, and for pre-processing we are going to use standard scalar for standardization and Schillhortz code for the measurement of the model training. First, we are going to import the dataset using read CSV, YNFCM dataset, and we are going to print first 5 rows of the dataset. Next, we are going to handle the missing values using mean. So, using fill now, we are going to handle the missing values where we are going to take the mean of the column and then we are going to place all the missing values with the mean which we have got. So, later, after enlarging this step, we are going to print the total missing values where we get 0. So all the missing values have been added. Next, we are going to standardize the data. Where, where after the standardization, now here we are going to see the standardization on a given dataset where features may have different scales, such as alcohol and acidity, or some of the columns, which is nothing but features in our dataset, where during the standardization, it makes mean from 0 and to the variance 1. So the mean and variance do not vary so much while we are doing the training and testing in the dataset. So SVM uses distance metrics, so scaling is important in the SVM. Next, we are going to initialize the learning rate sigma and maximum iterations for our model to be trained. So here we take learning rate as 0.5 and sigma function, which is nothing 1.0, and iterations will be 1000 iterations over the function. Next, we are going to visualize the input features which we have taken, and using matplotlib we are going to use scatterplot and then visualize the features which we have taken. So title would be feature distribution of the input features where scatterplot takes x-axis as alcohol and y-axis as malic acid and gives the visualization. So this is all. Next, we are going to perform SOM clustering with different number of clusters in group, where it helps uh, us to visualize how data points are distributed. We try different clusters from range 2 to 6, which is nothing but 2, 3, and 4, 5. So here we are going to try from different cluster sizes as told before 2 to 6, and now SOM maps different data into neurons which are nothing but clusters. Each data point is assigned to its winning neuron where Silhouette score measures the clustering quality. So first we are going to take the training of SOM each cluster. For example, if it is in the second cluster, it takes the second cluster, and at each iteration it will calculate using Minnesota library which have been important with the length x-axis as k, which is nothing but 1, and then we are going to take the sigma function as I've shown before, and then learning rate would be 0.1, 0.5 from the metric which we have assigned before. So each time it calculates the weight, each time it initializes its training model, it checks, and then if it is valid, it will it will get updated into the function. So at each and every time it gets loaded into labels. So after 4 iterations, the training of SOM would be 2, 3, 4, and 5, and each and every time after this it will get stored into labels in array function. At last we are going to print the Silhouette score. The Silhouette score would be 0.36 for cluster 2, 0.48 for cluster 3, 0.40 for cluster 2. So here Next, we are going to plot the graph for silhouette score, which has been calculated before for the range from 2 to 6, which is 2, 3, and 4, 5 clusters. From the score we got, as you can see here, 0.36 to 0.4, there is a variation, so the graph increases and then it decreases and then falls down. So next we are going to evaluate the performance of clustering model using silhouette score, which is also there are some more performance metrics which would be partition coefficient and partition entropy for different number of clusters. So in this we are going to take silhouette score, which measures the cohesion and separation, and partition coefficient, which compares the cluster compactness, and partition entropy, which would help in fuzziness and uncertainty. In experiment 7, where we have looked into the fuzzy means clustering, where it points out the data not just in one cluster but in the different clusters. So in the same way, we are going to measure the performance of this model using the principle of uncertainty. And in SOM, the mostly used performance metric would be silhouette. So looking into it, we are going to take two functions, partition coefficient, partition entropy, and we are going to formalize it. At each iteration, once after it starts from here, when the partition coefficient function is called, it goes to it and checks for it. So after each and every iteration, the cluster of 2 would get the silhouette score as 0.37, partition coefficient as 0.50, partition entropy as this. So after all this, at the very last step, before initializing or selecting the best model, we are going to first check for the optimal number of clusters which would have the high silhouette score. So for that, we are going to go to the iteration again for each and every cluster and check for the silhouette score. Which has more silhouette score, that model would be the best model. So for that, we are going to iterate, and the best model which we could get is cluster 3. So next we are going to display the centroids of the selected best model. So as from the big above thing, the best model would be the cluster 3, K3. So for that we are going to get the centroids. The centroid of those clusters will be minus 0.27 and so on as given here. Next we are going to visualize the points, cluster data points and the cluster centroids of the optimal number of clusters, which is nothing but best model we have derived from the above uh, generalization, assigning different colors to each cluster. So here we can see here we have got three centroids for the data points, and we can also see here the data points are slightly follows the other clusters and nearby to the other. So using scatter plot, we are going to plot this graph, and on the X label we have alcohol and the Y label we have malic acid. By deriving the above centroids and above transformation function, we will plot the graph. Now we are going to uh, send the output which have gone into the new file, which is nothing but ynfcm output.csv. In this file, the training and testing which we have done on the SOM would have been stored in this file. So now we are going to look the visualization in Power BI, how it varies from the Python program. So the datasheet which has been imported in the Jupyter Lab, the ynfcm.output.csv file, which is has been downloaded and imported into the Power BI notebook, and then it should be the output we will get for the scatter plot for plotting the graph between malic acid and alcohol for x and y axis. As you can see here, the clusters are pointed into three K1, K3 clusters. As we have seen before, the best model would be for the three clusters for SOM. The three clusters were plotted here using scatter plot. And here there are some more graphs are plotted using the same datasheet. And this would be the bar chart between the cluster and the sum of clusters. So if the cluster is 1, and it ranges from the sum of clusters to this. and for the count of alcohol by cluster to the cluster, and this would be the graphic. So this bar chart and page bar chart are pre-derived for our more informative. It should help us to plot more graphs in Power BI using the graphs which we are using. So this main aim of this experiment is to plot the scatter plot graph to visualize how these data points are plotted and which is also by using Power BI notebook. Thank you.